thank you all for coming to CU Boulder and for coming to the Department of Molecular, Cellular, and Developmental Biology, or we say MCDB is short for that. And my name's Lee Neiswander. I'm the chair of the Department of MCDB. And I'm really happy to welcome you to what we really hope is going to be the first in a series of community lectures here in Boulder, along with the Keystone Symposia, to really highlight important and timely discoveries in the life sciences. And so before I tell you a little bit about our department, I wanted to highlight this partnership that has been established with the Keystone Symposia for Molecular and Cellular Biology. And we were really excited when the Keystone folks came and asked whether we wanted to do this public lecture together. And then we were definitely blown away when we heard who our first speaker was going to be. You're going to hear from one of the top and most renowned scientists in the world right now. And you're going to hear about discoveries that are really the kind of technological advances that are really going to set us on a whole new trajectory and really is so impactful, probably the most impactful of this century. So let me tell you a little bit before we get into that about MCDB and who we are, where we came from, and a little preview of where we're going. So, okay, so the building that we're in now, Gold Biosciences, wasn't actually named after one of our CU colors. It's actually named after Professor Larry Gold. Larry Gold is, he's still part of MCDB. He was here from almost the beginning, and he's still an emeritus professor. And Professor Gold is a fellow of the National Academy of Innovators. And so during his time here at MCDB, he came up with all sorts of new inventions. He started a number of companies. One of the things that he did was to essentially make an evolution in a test tube in a method that we call CellX. And then that's been very important for the pharmaceutical industry. And he also developed a number of biotech companies, including Synergen, Nexogen, and Somalogic, which is just down the street. So the other main campus that we have, the other main building that we have on the campus is the Porter Biosciences building. And this is named after Dr. Keith Porter, who was the founding member of MCDB starting back in 1968. And so that was exactly 50 years ago. So we're turning 50 this year. We're really excited. Hope you guys will all stay tuned. We're going to have a number of celebrations this fall to celebrate our golden anniversary. So Keith Porter, just to tell you a little bit more about him, he pioneered the science of biological electron microscopy, which allowed us to peer deep into cells at a really high magnification, unprecedented levels. And he was the first to take a photo of a cell at this level of resolution on an electron microscope, and largely ushered in the modern field of cellular biology. He designed his own microtome in a way, this hand-cranked machine, which could make very, very thin tissue sections, 80 nanometers, and then could put that under this high-voltage electron microscope to get images. This microscope, which was in MCDB, was 2 stories high and one of only just a few in the world. So if you remember back to your high school days, you might remember hearing about the cellular structure that's called endoplasmic reticulum, or ER. And so what we see here is a mitochondria and another mitochondria here. But what you see is all these membrane structures that are studded with these little black dots. And those little black dots are ribosomes. They're our protein machinery. And this is an image from Dr. Porter that first allowed him to see and to name the ER. So over the past 50 years, MCDB has made many, many discoveries in cell and molecular biology and life sciences. But I wanted to just tell you a little bit of where we are now in MCDB. And I'm going to first come back to this ER, this endoplasmic reticulum. And so in this movie here, the ER is highlighted with a green stain. And Guy Volz's lab has discovered that the ER is essentially like the major trafficking center in the cell. And so these little red dots are cargo, cellular cargo, that's being trafficked along the ER to get to where it needs to go in the cell. And the ER, as the Volz lab also discovered, is important in also shaping organelles within the cell. And so in this case, this red long thing here is actually mitochondria, again, that powerhouse of the cell. And it's being enwrapped by the ER and then actually split into to make 2 mitochondria. So Ray Yi studies stem cells and how they control growth and development of important tissues such as the skin. And the Yi lab is discovering genes that when they go awry can give rise to disastrous consequences such as skin cancer. Joel Kroll is one of our newer scientists in MCDB, and he's been interested and discovered that bacteria, which are shown here, use electrical activity to communicate with one another, a lot like our neurons in our brains. And so in this picture, this is a group of bacteria that are sending out little electrical charges to one another. And they're actually being squished by jello, and so this is kind of their sense of touch that they are seeing here and giving this voltage. And then Zoe Donaldson is our newest faculty member. And so she's interested in understanding the neurobiology of complex social behavior, in particular that age-old question of what's love got to do with it? And so these little cute guys are prairie voles. So unlike most mammals, prairie voles actually mate for life. And so she's interested in using advanced technology to understand the neural circuits and the molecules that are involved in making these guys faithful over their lifespan. And so I hope you'll keep in touch with us and follow what's happening in MCDB. We're all very excited. And now I'd like to turn this over to the Keystone Symposia to tell you about the enormous impact that they've had on life science research since they started in the 1970s. And I'd like to introduce Thale Jarvis, who's the Chief Scientific Officer at the Keystone Symposia, to tell you about the mission and the vision of the Keystone Symposia.
Thank you, Lee, and welcome everyone. Keystone Symposia, in case you're not aware, is a nonprofit organization that's headquartered up in Silverthorne, Colorado. And like MCDB, we've been around for a while. We haven't quite hit 50 years, but we will in about 3 years. So we were founded in 1972. And our mission is really to catalyze the advancement of biomedical and life sciences by bringing together the key thought leaders in the life science fields at in-depth scientific conferences. And many of our conferences are held in Colorado, some of them at Keystone Resort, although I will point out that we are not in a business affiliate of Keystone Resort. We are a separate organization, a nonprofit. And the community lecture series is part of our effort to bring some of these thought leaders into the community and share some of the really exciting breakthrough discoveries that are happening in the life sciences right now, and give the community a taste of the type of science that is being discussed at our conferences. And we were very, very excited to bring Dr. Feng Zhang to Boulder today to share some of his work. I actually have a long history of working in Boulder. I actually was in this department, MCDB. I think I remember MCDB celebrating its 25th anniversary. I came here as a postdoc in Carla Kierkegaard's lab. This particular room, I think, didn't exist when I was a postdoc, but the department has expanded, and it's really actually fascinating to see what some of the new faculty are doing here. But anyway, happy to be back in Boulder. I've been the Chief Scientific Officer at Keystone for about a year. And we're, as Lee mentioned, we're really hoping that we can make this community lecture series a regular event, bringing it to Boulder. So I'd like to introduce Feng Zhang. He is a really eminent scientist. He's a molecular biologist that's focused on better understanding brain function and trying to seek cures for neurological diseases and psychiatric diseases. He has been a real pioneer in the area of genome engineering, and actually, so he started out getting his undergraduate degree at Harvard University, where he got a bachelor's in chemistry and physics in 2004. And then he went on to Stanford University, where he worked with Carl Dieseroff's group. And he actually made some very key contributions to an emerging area called optogenetics. And I don't know if we're going to hear anything about optogenetics today, but that's one of his other pioneering areas that his lab is still working on. He got his PhD in 2009 from Stanford. That was in chemistry and biophysics. And then the Boston area was quick to snatch him up again. He did a short fellowship at Harvard and then went on to the Broad Institute of Harvard and MIT, where he currently is a faculty member. So he published a paper in early 2013 in Science, which is considered one of the really key papers in precision genome engineering. It was the first successful work in genome editing in mammalian cells, and that it is one of the most cited papers in genome editing. His lab has since gone on to further refine that methodology. That the 2013 paper was on CRISPR-Cas9 technology. He's since gone on to develop a technology that they're calling CPF1, which is a more refined methodology for editing DNA. And even more recently, his lab has worked on a system called C2C2, which is for editing RNA. So it's a great pleasure to welcome him for this lecture. As I mentioned, he's a core member of the Broad Institute of MIT and Harvard. He's also an investigator in the McGovern Institute of Brain Research at MIT. He's the James and Patricia Poitras Professor of Neuroscience at MIT. He's an associate professor in the departments of Brain and Cognitive Sciences and Biological Engineering at MIT. And he's also a Robertson investigator at the New York Stem Cell Foundation. So it's my great pleasure to welcome Feng Zhang.
First of all, thank you so much for the kind introduction, but even more so, thank you for coming out here on Sunday afternoon. You must have better things to do than sitting here and listening to my talk. It's really a great pleasure to be here, and I wanted to share with you some of the work that we have been working on in developing genome editing technologies to be able to make very defined changes in the DNA of our cells. There are a lot of different potential applications that range from advancing research to developing therapeutics to engineering crops so that they are drought resistant or virus resistant so that we can improve agricultural yield. And also, I will tell you about some of the ongoing activities of developing new technologies that have applications for diagnostics and also continued development of therapeutic strategies for treating diseases. So we'll get started with this slide here. So over on the right, this is the plot from the National Institute of Health. And what you see here on the y-axis are the number of papers that have been published year by year over the past decade. Each paper here reports a series of discoveries that link genetic differences to specific diseases. Now, this is really still at the very tip of the iceberg, because as we start to sequence more and more human genomes, we'll be able to identify many more genetic differences that are linked to specific disease. So some of these genetic differences play what we call a causal role in the disease. So that means if someone has a mutation, then they will have a specific form of illness. And so of those causative genetic mutations, there are about 10,000 that have been identified to date. And so if you think about that, if we know what mutation causes disease, then you might ask the question, why don't we just go into our cells and get rid of them? If we know what is a deleterious mutation, let's go into the cells, change it so that we can reverse it back to the healthy DNA sequence. It turns out that to do that is fairly challenging, and that's one of the challenges that both my lab and also labs around the world have been working on to try to advance. So the simple idea of how to go about changing DNA is here in this picture. So before I explain to you what this picture means, the human genome has 3 billion letters long. And so, and we have twice of that. So we actually have 6 billion letters because there are 2 copies of each DNA in our cell. And so a mutation in the DNA can happen anywhere in this 3 billion letters. And so if you think of the genome as a document of 3 billion letters, then we need a way to be able to search in this document, find where that mutation is, that typo in the document, and then be able to reverse it so that we can correct it and restore the healthy function of the cell. How do we do that? How do we search for it? Turns out nature has invented a number of different ways to be able to search along pieces of DNA. And these systems are called DNA binding proteins. By the name, you already see that their function is to interact, bind to a specific DNA sequence. So the basic idea for doing genome editing is this. If we can have a DNA binding protein, a DNA binding domain that we can reprogram, customize it to recognize, search for a specific DNA sequence, and if we link it to a protein called the nuclease, then we can achieve genome editing. What these nucleases do is that they can introduce a double-stranded break, a cut in the DNA. And so more than 15 years ago, Gene Haber and also Maria Jason, 2 eminent scientists in the field of DNA repair, discovered that whenever there is a DNA break in the genome, that break triggers DNA repair machineries and allows the cell to repair that DNA break with much higher levels of efficiency. And so a lot of the work have really focused on how do we reprogram this DNA binding domain with significant ease so that we can, for any sequence in the genome, quickly and efficiently develop a DNA binding protein to find that sequence in the DNA and to be able to manipulate it. So some of the earlier technologies to be able to search in the genome include zinc finger proteins and also tail proteins. These are protein-based DNA binding domains. And what that means is for a zinc finger protein, there are different modules. Each one of these colored sort of squarish things is one module of a zinc finger protein. Each one of these naturally recognizes 3 DNA bases. And you can mutate these proteins to get them to recognize a different set of 3 DNA letters. When you do that, then you can link them together to form what we call a zinc finger array. So it's simply a tethered system with multiple fingers. And if each one recognizes 3 bases, then a zinc finger array can recognize a multiple of 3 bases based on the number of modules you have in it. So for a 3-module zinc finger, you can recognize 9 DNA bases. And for a 6-module, you can recognize 18 DNA bases. But even though this paper looks remarkably simple on paper, it turned out it's actually not so easy to use. And why is that? That's because if we want to change the sequence that this module recognizes, we'll have to change the protein. And the way that a protein folds into a specific shape to be able to recognize the DNA depends on the interaction of many of the residues. So when you change one, it affects many other ones that this module makes contact with. And so the problem sort of becomes exponentially harder because when you tweak one, it changes other things, and so it's not very predictable. And so as a result, to build a new zinc finger protein that works well, it can take several weeks to months. And even then, you're not guaranteed to develop a new zinc finger that works well. So a system that's a little easier than zinc finger is this tail protein. Here, is simpler because each module recognizes a single DNA base. And so that means you only need 4 modules rather than having to have 64 modules. So this system is easier, but still is hard because the way that each module recognizes a base is affected by the neighboring modules. And also, it's just a cumbersome process to be able to engineer one of these proteins. So we were working on both of these systems, but because they were challenging, I thought, can we come up with a simpler system? And it turned out that nature has, in fact, invented other ways to be able to target DNA with remarkable ease. And that system is something that's called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeat, but I'm going to just call it CRISPR. Otherwise, the talk will take forever to finish. So what CRISPR is an immune system in bacterial cells. Just like we have our own immune system, our immune system uses proteins, antibodies, to recognize invading viruses or bacteria or other things. CRISPR uses RNA to recognize invading viruses. So if you think of this gray box as a bacterial cell, when a virus infects the bacterial cell, it injects its own genetic information, usually in the form of DNA or RNA, into that bacterial cell. So once the virus has injected itself into the bacteria, then this system called CRISPR, which is encoded in the genome of that bacteria, gets activated. And there are genes that encode CRISPR-associated proteins, which are called Cas genes, that get turned on and they start to become these CRISPR proteins. And these proteins working together with RNA that are derived from this repeat element called CRISPR array acts as a surveillance complex for recognizing that invading virus. So here is one example of one of these surveillance complexes. There's a protein called Cas9, and then Cas9 works with a short RNA that we call a single guide RNA. And so this single guide RNA is an engineered form of these natural CRISPR RNAs to be able to recognize the invading virus DNA. And so wherever this RNA matches with the DNA, so when the blue virus DNA matches with the red guide RNA, then the Cas9 enzyme will make a double-stranded cut in the DNA so that it can inactivate the virus DNA. But this same mechanism of searching for DNA sequence and then making a cut is exactly what we may be able to use for doing genome editing. And so what we have been working on then is trying to see, can we harness this system, engineer it so that we can use it to edit specific sequences in human DNA so that we can achieve genome editing. So I got started with this work when I first read Sylvain Moynou's paper back in 2010, where he reported that wherever you have the RNA sequence, which is highlighted here in green, pairing with the target DNA sequence, which is in black, if there is perfect recognition indicated by these vertical black bars, then the Cas9 enzyme will make a double-stranded cut within the DNA, so exactly right here. And this double-stranded cut is the end of that reaction. This enzyme doesn't go and start to make more cuts in the virus or in the plasmid DNA. And so this is a very nice feature because this shows that you can program Cas9 with this RNA sequence to make a precise double-strand break. And that suggests if we can harness the system and make it so that we can program this enzyme to recognize a specific human sequence, then we can go and cleave at a specific location in the genome and then stimulate DNA gene editing. And so working with a number of graduate students, including Le and also Anne, we successfully introduced the Cas9 enzyme as well as engineered the guide RNA so that you can recognize the target DNA in the human genome and then introduce the double-strand break. And when that happens, they initiate the repair processes so that we can go and either delete sequences or incorporate new sequences in the genome. So subsequent to that, we then also, working with additional members of the lab, started to interrogate the specificity of the system. So how precisely can we use this enzyme to be able to edit a specific sequence in the genome? And then also at the same time, developing computational tools that allow researchers around the world to be able to easily design guide RNA sequences so that they can target their respective genes of interest. So I've said a lot, but a picture or a video is worth 1,000 words. So here is a video that shows you how the CRISPR-Cas9 system works. So in blue here, this is the Cas9 protein, and then in red is the RNA that we programmed to recognize a specific sequence in the human genome. So by introducing this complex through the human nucleus membrane and then getting it to recognize a specific sequence in the human genome, when the RNA, when the protein opens up the DNA, which is in blue, and then when the RNA base pairs with that DNA to achieve recognition, then Cas9 makes a double-stranded break so that we can stimulate DNA repair. So there are two ways to repair the DNA. One of the ways is simply rejoins the two ends of DNA together. But the process of achieving this introduces a small mutation, and this is quite useful for inactivating a gene of interest. Sometimes there are genes that are deleterious, and we can use that to inactivate it. The second way to repair DNA is more precise. So here you have the two broken DNA ends. You can provide the cell with a new piece of DNA that is similar to the two broken ends, but carrying a new sequence that you want to introduce into the DNA. This could be a sequence that you want to swap into the genome to replace the mutation. And the repair machineries will incorporate this directly into the genome. And so this video shows that there are two ways that we can go and edit the genome. The first way is to make a cut, allow the cells to re-glue the ends together to make a inactivating mutation that gets rid of a deleterious gene. And second, you can give it a template DNA that allows you to switch in a specific sequence of interest in the genome. So that's how we go about doing genome editing. So in addition to editing the genome, turns out that these systems also have additional applications. And so the first application that the video describes is using it as a nuclease to cleave DNA and then stimulate DNA repair. But then the second class of application is using this Cas9 protein as a way to bind to specific DNA sequences so that we can bring other functionalities to a specific location on the DNA so that we can start to manipulate it. And this is done by getting rid of these two domains on the Cas9 protein that naturally are responsible for cleaving DNA. So if we get rid of them, then Cas9 will simply bind to DNA and not cleave it. And then we can use this binding protein to bring effectors, like things that can turn on transcription, which allow us to turn the gene on, or turn off transcription by closing the chromatin, condensing the DNA. Or we can bring in a fluorescent protein like GFP so that we can visualize the configuration of a specific location in the genome so that we can track its motions as the cell is undergoing some type of biological function. And there are many other ways to use this basic system. And these are all ways that biologists now are using to advance our understanding of different biological processes. So following those initial work to develop the CRISPR-Cas9 system for gene editing, we thought to further enhance the system. And so one of the really exciting potential application of CRISPR-Cas9 is to use it as a therapy. If we can deliver that into cells in specific tissue, correct genetic mutations, and then achieve a therapeutic outcome so that we can treat disease. But one of the hurdles for enabling that goal is how do we deliver the Cas9 protein into the right tissue, into the right cell efficiently. So one of the challenge that faces delivery is that the Cas9 that we initially developed is quite large. It's hard to fit it into delivery systems that we can use for delivering into, say, cells in a mouse. And so working with members of my lab, we examined the diversity of Cas9. So it turns out that Cas9 is a bacterial protein, and many bacterial cells carry their own version of Cas9. And they come in all shapes and sizes. Some are larger, some are smaller. And the first one that we developed was pretty large. It's from the Streptococcus pyogenes bacteria. And then we also developed a smaller one from Streptococcus thermophilus, but that enzyme is not as efficient as the S-pyogenes Cas9. So we thought maybe let's examine this group over here, the smaller bundle of Cas9. Some of them are smaller, and if we can find one that works well, then we can more efficiently achieve gene editing in animal models. And so we examined a number of these different small Cas9s, and we eventually found a specific one, and that's the SA Cas9. So SA stands for Staphylococcus aureus, which is another human microbiome bacteria. And so by taking this small Cas9, we can introduce it into a viral vector. Viral vectors are what we call gene delivery systems that people use for gene therapy, for transferring genetic information into specific human cells. So we can package the genetic information from Cas9 into this virus, along with the RNA guide that we use to target a specific gene of interest. And so this allows us to produce viruses, deliver Cas9 into a mouse model, and then see, can we achieve efficient or sufficient level of gene editing to achieve a therapeutic outcome. So to test it, we designed a guide to target the gene PCSK9. PCSK9 is a prominent therapeutic target because there's a small population of individuals who naturally don't have PCSK9. And they live long and healthy lives, and on top of that, they have very low levels of cholesterol, so they don't have high levels of heart disease risk. And so drug companies have been developing either small molecules, so chemicals, or proteins to be able to deplete PCSK9 from human patients who have high risk for heart disease by reducing their cholesterol level. And so we thought if we can use Cas9 guided by a RNA that targets the PCSK9 gene to inactivate PCSK9 permanently, then we may be able to achieve a long-term treatment for cardiovascular disease by mimicking this natural mutation that's found in some individuals that have low heart disease risk. So here is what the data looks like. So the very left panel shows gene editing as measured in the mouse that we injected. And so these are different days after we injected the virus into this mouse. And you can see we achieve about 50% editing in the liver cells of these mice. We target the liver because that's where this PCSK9 protein is produced. And so we were primarily interested in editing hepatocytes in the human liver. So once we had that, then we thought, if we're getting 50% editing, that's pretty good. Does this translate into some kind of decrease in the PCSK9 protein level? So here, this plot, we measure the amount of PCSK9 in the serum of these mice. And what we find is that corresponding to the timeline here, 7 days after injection, PCSK9 became undetectable in the blood of these mice. And comparing this with the control, which is here in the gray line, you can see that there is a very drastic decrease in the amount of PCSK9 in these animals. So if you reduce PCSK9, then we should also be reducing cholesterol level in the mouse. And so that's what the right figure shows. This is measuring the amount of cholesterol in the serum of these mice. And what we see is also after 7 days, all of the experimental mice that received the gene editing reagent reduced cholesterol by 50% compared to the control animals. And so when you can reduce cholesterol by 50%, that's a significant advantage in terms of susceptibility to cardiovascular disease. So this shows, it's promising, but there are still a number of things that we need to do to continue to develop the system so that we can have efficient and also precise and also safe gene editing to be able to achieve therapeutic outcome in patients. So a lot of work is going on now to, one, achieve more efficient delivery, and two, making sure that the system is safe and efficient so that when we inject it into a human patient, we can achieve the desired outcome. So one of the things that we have realized as we were developing the Cas9 technology is that there likely will be many other technologies out there that nature has invented. And if we can go and harness the natural diversity that exists in the bacteria that live around us, we may be able to find other interesting systems that are suitable for advancing human health. And so one thing that we have been doing for the past few years to explore bacterial diversity, all of these different microbes that live around us on our body, in our environment, and also even in extreme environments that we don't live in. And so some of the work we've been working on in this front has been to see, are there additional CRISPR-Cas systems that we can harness to be able to advance the gene editing technology. And so what we did is we first examined the way that CRISPR-Cas9 systems are present in bacterial cells. Turns out that there are two major classes of CRISPR systems. There are the class 1 systems that use many different protein modules that work together to form a big, complicated machine to perform the recognition and destruction of invading viruses in bacterial cells. And then there are the simpler systems, which are the class 2 CRISPR systems. These have a single protein, which is a much simpler machine to be able to recognize the invading virus and also destroy it. At the time of our study, there were many different class 1 systems, many complicated systems that scientists have discovered, but only one simple system, and that's the Cas9 system that had been reported. So we thought, if there are many class 1 systems, who's to say that nature didn't also invent additional simple class 2 systems to be able to provide bacterial defense? And so working with our colleague Eugene Koonin at the NIH, and also members of my lab, we set out to perform a computational investigation to see, can we identify new, novel, simple CRISPR systems that will maybe the harness for achieving gene editing. And so by developing a computational pipeline that uses some of the most well-conserved elements of a CRISPR system to be able to identify other CRISPR systems that people haven't yet identified, we then were able to derive a number of candidate genes that we then followed up to develop biotech applications. And here is a summary of some of these CRISPR systems that have been identified to date. Chiefly, there are three major groups of CRISPR or single component CRISPR systems. There are the Cas9s, which are the gene editing system that I told you about. And then there are the Cas12s, which are also DNA editing enzymes. These are systems that include CPF1. And most interestingly, in addition to these two DNA targeting systems, we also found that there are RNA targeting systems that are part of this Cas13 family of CRISPR systems. So without telling you more about the DNA systems, I'll focus probably the rest of my talk on this RNA system that's called Cas13. It was previously called C2C2, but just to make sure that everything lives in a happy CRISPR family, we renamed it to be Cas13. So to figure out what Cas13 does, we took the Cas13 system from a bacteria that's pretty challenging to study and transferred it into E. coli. We like E. coli because it's easy to manipulate in the laboratory, and we have a lot of tools that allow us to study it. And so once we transferred this into E. coli, we then did RNA sequencing to make sure, to see whether or not these CRISPR RNA guides are expressed. And we found that indeed they are, which is a very good sign. It suggests that this is a system, even after transplanting into E. coli, is likely still functional. So based on the hypothesis that this is an RNA targeting protein, we then set up a screen to see, can we validate that this is an RNA targeting system? And two, can we learn what is the rule that the system uses to be able to search for RNA and also target it? And so to test this, we chose a bacterial virus called MS2 phage. And this is a virus that only exists in RNA form. And so we chose it specifically because if we chose a virus that also exists in DNA form, then when this Cas13 system shows defense of that virus, we don't know whether or not it defended the DNA version of that virus or it defended against the RNA version of that virus. But if we choose a RNA-only form, then if there's any defense activity that we observe, then we know it's defending against RNA. And that means Cas13 is targeting RNA and not DNA. And so the way we do this is we designed RNA guides that tile the entire length of this RNA virus genome. And that way we can cover the entire virus. We didn't know what are the rules that govern which sequence to target and which one to be not targetable. So by tiling that, we basically saturate the entire virus so that something should work if it does work. And so we synthesize all of these RNA guides using inkjet oligo printing technology, and then cloned it into a library of DNA so we can introduce that into E. coli, and then generate a library of E. coli where each cell carries a different guide sequence targeting a different position on this virus. So now, by exposing this group of bacterial cells to this virus, if any of these RNA guides was able to defend the bacteria from the virus, then that cell will survive. And so by looking at the surviving cells and sequencing the guide that is left over in those surviving cells, then we can start to infer the rules that govern targeting of different virus sequences. So what we found is that indeed, some cells were able to survive, which shows that it is able to target RNA. And two, by looking at all of the RNA guides that provided a defense function, we found that there's a simple rule that governs targeting, which is that right next to the site on the virus that's targeted, it just needs to be flanked by a single base, and that's either a C or A or a U. And that makes the system quite easy to design and to be able to target in bacterial cells. So that's targeting the targeted sequence on the virus. And what we found is that in addition to targeting the targeted RNA, there's also a very interesting property we call the collateral activity. And what this activity is that if in a reaction you have the RNA that you're trying to target, but also the guide RNA, which recognizes the targeted RNA, if both of these pieces are present, then the Cas13 enzyme will get turned on so that it will go and cleave any other RNA that's within the same reaction. And so even if you have another piece of RNA that is not at all recognizable by the guide RNA, as long as this recognition happened, then this RNA will also get destroyed. So in bacterial, in the life of the bacteria, we think that this provides a very unique advantage for defending against very strong pressures of infection. But biochemically, this suggests that Cas13 operates in a different way as the DNA targeting systems, because in addition to cleaving the target, it also remains active and cleaves other RNAs in the same reaction. And so from this gel, the first 8 lanes simply show that as long as the targeted RNA is present, then the RNA that is not being targeted can also get cleaved, which are these different fragments here. But if you don't give the targeted RNA, so only the collateral RNA and also the guide RNA, then there's no cleavage. And this shows that it's a recognition-triggered RNase activity that goes and chews everything else. So you may ask, how does this fit in the overall context of bacterial defense? And this summarizes our understanding to date. So with the C2C2 or Cas13 system, what we think is that there are 2 modes of immunity that this bacterial CRISPR system is able to provide. The first mode of immunity is the standard immunity, which is when the virus infects the bacterial cell, Cas13 gets activated and destroys that virus, and then the bacterial cell can live on happily ever after. But if the virus is somehow able to evade the initial immunity and start to replicate itself, then we're in a much more dangerous zone. Because if that virus is able to replicate and spread to the rest of the population, then you end up with an exponentially amplifying infection activity. And so what Cas13 does is that if it recognizes that somehow evaded the immunity defense, then it will trigger what we call programmed cell death. And that's this RNA-triggered, recognition-triggered destruction function of all of the other RNA so that it can destroy all of the virus RNA through programmed cell death before this virus gets released into the community. So that single bacterial cell that gets infected destroys itself and protects the population. It's quite altruistic for this system to behave. So this is all very interesting microbiology, and you may be sitting there and asking, what does this have to do with improving human health, and how can we use it to make our lives better? Turns out that there are several different ways to apply the system, and the first application is that we can use the collateral activity of Cas13 to be able to develop a highly sensitive and also highly easy to use diagnostics platform for either pathogenic viruses and bacteria or for detecting cancer DNA. So the way this works is a system that we call SHERLOCK that basically takes as input biological samples, usually in the form of urine, saliva, or blood, and we can extract DNA or RNA from this biological sample. By performing T7 transcription, we can convert either the DNA input or the RNA input into a big batch of RNA. And then we can program Cas13 to recognize the virus or the bacteria we're trying to detect. So say we're trying to develop a Zika virus or a influenza virus detection assay, we simply program the Cas13 enzyme with an RNA guide that recognizes the influenza or the Zika virus sequence. And by incorporating this Cas13 enzyme and the programmed guide along with a reporter, that's just a short RNA with a quencher and a fluorophore attached together. So when they're attached, when this RNA is intact, the quencher quenches the fluorescence, so you don't see a color from this reaction. But if the Cas13 molecule, along with the guide that programs it to recognize the influenza virus or the Zika virus, when that recognizes the virus sequence, then it will trigger the Cas13 enzyme to become activated so that it can go and cleave these reporter RNA as well. And so that's when you get the quencher released from the fluorophore, and you get a fluorescent signal from this reaction. So using this, you can get several orders of magnitude amplification. You get amplification here and also amplification there, so that we can detect molecules with sensitivity as low as 1 molecule per microliter, which is the level of sensitivity you need to be able to detect early stages of virus infection. So this is how the system works. Turns out that the Cas13 enzyme is very robust. You can freeze dry this onto a piece of filter paper, and then you can apply the biological sample onto it and then read out the fluorescence. And so using this freeze-dried format, we show that you can detect as low as about 20 nanomolar. And so that's about 10 molecules per microliter of the material they're trying to detect. And so this is already much more sensitive than, say, the over-the-counter HIV detection kits that you can buy from the store. So that was for detecting viruses. You can also detect the DNA. And so over here, with these two different plots, what we show is that you can easily program the system with a guide that recognizes E. coli, by giving a E. coli genomic DNA or giving a Pseudomonas genomic DNA. And you can program very specifically a guide that recognizes it with the E. coli sequence. But if you give it guides that recognize other bacterial cells, you will not get a signal. And this just shows that you can very accurately and also easily program the system to recognize different infectious material. So that was all done using fluorescence-based readout. But it would be really nice if we can also have a colorimetric readout so that we can use this in the field, so that we don't have a fancy fluorimeter to be able to read whether or not the virus is present. And so along that direction, we've been developing a lateral flow-based method where you can apply the sample to a paper strip, very much like a pregnancy test strip. And you can apply the sample, let it flow through the strip, and if what you're trying to find is present, you will get a new band appear in this paper strip. And so for this lateral flow test, we developed using the Cas13A system, and we can get a pretty good detection of Zika virus RNA. And so these are different strips where we applied a Zika virus sample, and you can see we can get a pretty sensitive detection using this paper-based test. So using this and further developing the sensitivity of this lateral flow system, then it's possible to have over-the-counter or even at-home diagnostics applications for a variety of different diseases. This will be especially useful because once a person suspects that he or she has contracted an infectious disease, the last thing you want that person to do is to go out into the subway, travel to the hospital, spread the virus everywhere that person travels. It's much better if the person can just test it at home. And if that is the case, call 911 and then apply quarantine procedures so that we don't have a public health hazard in this case. So that's something that we're working on to be able to make it easier to be able to detect diseases. So the second application of using this Cas13 enzyme is to use it to edit RNA. So earlier I told you about editing DNA, but turns out that there are a lot of benefits to be able to edit RNA. And so one of the benefits is that the DNA nuclease-based genome editing is not very efficient in cells that don't replicate. In fact, many of the cells in our body no longer replicate. If they were replicating, we would be growing taller and taller and bigger. And so to have effective therapeutic efficiency, we need to overcome this challenge. And this is one thing that editing RNA allows us to overcome. The second thing is that RNA editing provides reversible modification of DNA. A reversible modification of the cellular property. One of the concerns about DNA editing is that if it does go and make an off-target edit, so something that we don't want to edit, those DNA edits are permanent. But when we edit RNA, we can overcome that particular consideration. So RNA editing naturally occurs in cells, but what doesn't happen naturally is that it's hard to reprogram it to edit other RNA sequences. And so by developing Cas13, we now have an easy homing device that we can link to an RNA editing complex so that we can direct it to edit any RNA sequence we want. And so the way we do this is we begin with the Cas13 protein, just like how researchers inactivated the Cas9 nucleases so that it only binds to DNA. We inactivate the RNA's domain so that it will bind to RNA but not cleave RNA. And so once we have this, we have a RNA binding or RNA homing device that we can use to bring RNA editing enzymes to a specific location. And so this is what we did. We have this inactive or dead Cas13, and we use it to bring the RNA editing enzyme ADAR to a specific sequence. And then we can program which RNA base to change by mispairing that RNA base with a particular cytosine on the guide RNA, and you can achieve very precise adenosine to inosine changes. So inosines are read out by the ribosome as guanosine, so effectively we're making A to G changes in the cell. And so this is just a more technical diagram showing you that if you have an RNA on top that you're trying to change this particular adenosine, you can design an RNA guide where you mispair the adenosine with the cytosine, and that specifies which one to change. And so when you do that, even though there are other adenosines that are also bound by Cas13, because they're not indicated with the mismatch, those are not edited at the same time. And so we tested the system. There are two different ADAR proteins in the human proteome. There's ADAR1 and ADAR2. Both of them can mediate editing. ADAR2 is a bit more active, and so we chose to focus on ADAR2. And so one of the really important things to look at is how specific is this RNA editing system. And so this yellow dot is what we want to edit, but unfortunately, when we sequence the whole transcriptome, so every single all of the RNAs in the cell, we'll find that there are a whole bunch of other sites that are also edited, which is very disappointing. But we thought to figure out what's actually happening, and we did that by performing a control experiment where we have a guide that doesn't target what we're trying to target. And when we did that, we found that many, if not most of the off-target overlapped with the off-target sites with the targeting guide. And that suggests it's probably not, these off-targets are probably not indicated by the RNA guide, but rather probably by the ADAR enzyme or the Cas13 enzyme itself. And so we thought, based on this understanding, we can maybe engineer the system to make it more precise. So this is repair V1, and you see there are about 2,000 off-target sites. And so by looking at the crystal structure, which was solved by Peter Beal's group at UC Davis, we were able to identify different residues that contact the RNA. And then by mutating these different residues, we thought maybe we can increase the specificity of this RNA editing system. And we tested a large number of different mutations. Some of them, like the T375G, seem to dramatically increase the specificity. This is measured using a specificity score. And so we thought this is interesting. Maybe we can test this to see does it make the RNA editing system a lot more specific. So to test this, we did this experiment where using the repair V1, you not only see editing of the targeted base, but you also see editing in other locations within the target site. But when we engineered with the T375G mutation to make the repair V2 system, we are now able to significantly clean up the editing event. So you only see the targeted base edited, but not the other non-targeted bases. And then we did more experiments. So now we sequenced the whole transcriptome at 125 times of coverage. This is much higher than what people do typically for RNA sequencing. And so now, because we sequence it higher, we now see about 20,000 off-target sites with the repair V1 system. And then just for comparison, this is the repair V2 system. And you can see now we have 125x reduction in the number of off-target sites. And so working on this, working in this direction, we can make the system a lot more precise. And one thing we're working on now is trying to bring this on-target activity back up so that it has the same level of activity as the repair V1. So decreasing off-target, increasing increasing activity. So we're continuing to develop these gene editing systems. There are a lot of things to do. How do we increase specificity and efficiency? How do we deliver this effectively to different organs so that we can get closer to developing them as therapeutics? And then can we make other forms of RNA-based changes, not just A to I, but G to A or C to U, U to C, all sorts of other changes. And then evaluate this. How does it work? How well does it work in animal models? So, so that's what I have prepared for the data today. But one thing that we're continuing to look at is to continue and mine the bacterial and natural diversity that surround us. This is a plot that shows the number of genomes that have been completed, bacterial genomes that have been completely sequenced. And so what this really highlights is that what we are looking at now is likely only the tip of the iceberg. There are probably a lot of other interesting systems that remain that are waiting for us to discover. And as we study them, we'll probably be able to develop more powerful biological tools. So we're continuing to develop these technologies, but at the same time, as the technologies become more mature, we're now starting to apply them to both study and also develop ways that we may be able to treat neurological diseases, including Alzheimer's and also a lot of the psychiatric diseases. So that's all just want to, this is really the most important slide. I want to thank my group at MIT and Broad who worked with me, and also our collaborators, and also the funding agencies that support our work over the past 7-8 years. Thank you so much for your attention. So I'm happy to answer any questions you may have.
So looking at your Cas13 system, do you know what's actually structurally happening to the enzyme to make it a non-specific RNA degrader? And then is there, on the flip side, an off switch that you'll see to return to homeostasis?
Very good question. So, so the question really is, what is happening? Why is Cas13 able to stay on and cleave other things while maybe Cas9 and CPF1 don't? So I think part of the answer has to do with the way it cleaves the target. When Cas13 cleaves the target, when Cas13 recognizes a piece of RNA and cleaves it, it doesn't cleave in the recognition site. So it cleaves outside the recognition site. And so what that means is the guide RNA and the target RNA binding, that duplex, remains intact. And that's what Cas13 needs to be able to stay in this active conformation to be able to act as RNAs. In contrast, Cas9 or CPF1, they cleave in the recognition site. And so that means upon cleavage, then the target is destroyed. And so because it's destroyed, this duplex that activates the protein into a DNase is no longer there. So it gets silenced. Molecularly, we don't have the atomic structure for Cas13 yet. So we don't really know, how are the domains moving together to form this RNAs. But we're working on that, and hopefully we'll have the detailed atomic structure soon. Yeah. And I think you had a question.
So just last week, I was reading that some of the Americans who want to do clinical trials are just beginning to get down the pathway of getting approval.
Mm-hmm.
But China's been at this for about since 2015.
Mm-hmm.
Do we know anything about any of the outcomes of those clinical trials that have been going on for a few years?
Yeah, that's a good question. I have the same question, actually. So there are a number of clinical trials going on around the world. There's work happening in China. There's a lot of work happening in the U.S. At UPenn, in various companies focused on developing CRISPR-based therapeutics, and so forth. Unfortunately, we don't have very much information about what has been done and what the readouts are from the trials happening in China. What I know, based on reading, is that they are developing some type of cancer therapeutic using CRISPR to engineer immune cells so that you can put those engineered immune cells back into cancer patients to be able to target lung cancer. That's one of the trials that I read about in the published press. Beyond that, I don't think they have released any data. In the US, there are a number of groups working on developing CRISPR-based therapies also for treating cancer, probably mostly lymphoma for cancer, but also for sickle cell disease, for blindness, and also for some metabolic diseases that happen in the liver. I think there's a lot of progress being made, and different groups are filing for clinical trial applications in Europe or in the US. So hopefully in the next year or so, we'll start to get some information about how well these technologies are working from a clinical perspective. Yeah. Yeah. Uh-huh. Yeah, so one of the things that we have been working on is to use Cas13 for liquid biopsy. So liquid biopsy is where you simply take blood from a patient, and by reading or detecting the cell-free DNA or RNA that's circulating in the blood, you can make a determination of the disease state. So a lot of groups are working on looking at these cell-free DNA for cancer to monitor and also track the progression of tumor development in the body. We have been working on trying to develop Cas13 as a diagnostics method to read out are there tumorigenic DNA that's in the cell-free DNA fraction, and can we achieve quantitative readouts so that we can not only detect it but also measure it and track the growth of the tumor. That's one of the applications that you might be able to use Cas13 for.
I have a couple questions. So
Sure.
the first one is looking through the microbial diversity.
Mm-hmm.
What else do you think you might find? So for instance, do you think you might find protein editing systems because they're proteinaceous infectious particles like prions, right?
Yeah.
So that's one question. And then the kind of related, when you did the bioinformatics analysis, what features about the other bacteria suggested that it was going to edit RNA?
Right. Really great questions. So the first question is, if you look into this microbial well, what else can you find? I think you can potentially find anything. Nature is pretty amazing, and nature has had a long time to iterate and evolve and refine the function of all sorts of proteins, take them apart, put them together, mix and match domains from different proteins to form new proteins of new function. I think it's very likely that we'll find other proteins that may have interesting biotechnology application beyond just DNA or RNA cutting enzymes. There may be protein binding enzymes, there may be protein cutting enzymes, there may be sugar binding enzymes, there may be sugar cutting enzymes, things that can synthesize complicated chains of sugar molecules for cell surface, recognition or communication, things that manipulate lipid. I think it's really a rich well that we need to continue to explore. The second question, and one more point about the first question is that, and what is really exciting is that based on these hypotheses of what might be a protein that can perform a desired function, you can then start to mine the sequence databases. Look at all of the bacteria species that have been sequenced, look at their protein sequences, and then see does any of them conform to the hypothesis. And if it does, then you can take it to the next step and start to study it. So the second question is, when we were searching for CRISPR systems, what are the features that told us that it may be an RNA targeting enzyme? So that's when we found these proteins, actually this whole family of proteins that are fairly well conserved. We then examined these proteins, members of the same family, to see what are the different domains that are most conserved. If within a whole family of protein there is a domain that's highly conserved, then that domain suggests a common function that's preserved throughout the whole family. The whole family. And so two of the domains that are highly conserved are the happen domains. It stands for higher eukaryotic prokaryotic nuclease domain. And they are normally found in RNases or bacterial toxins. And that's what told us that this might be RNA targeting enzyme. So then based on that hypothesis, we can design a screen using the MS2 RNA virus and see does it target RNA.
You sort of touched on this in the beginnings. I was just, so for like diseases that that are undetectable early in life, how long does it take for those effects to be reversed?
Can you give me an example of what disease you might be thinking about?
Maybe like a later stage progeria that doesn't really affect people early on in life, but maybe later down the line.
Yeah. So I know about progeria, but I don't know that much about. I don't know a lot about late stage progeria. Is that caused by the lemon?
Yeah, the laminate
mutation?
protein.
Okay. So a lot of that, I think, the best way to do it is probably to detect by sequencing the genome, or running a genetic panel to see does the person carry that mutation. That's probably the best way to go. In terms of how quickly, say you edited the mutated gene in the patient, how long that will reverse the phenotype. For progeria, I don't know. I think because it's a, so I guess we can test in animal models to see is it a reversible phenotype. Some genetic diseases may be reversible, others may not be as easily reversible. So I think it will probably differ case by case. And some diseases can probably reverse much more quickly. For a blindness disorder where there's a mutation that affects photoreception, you might be able to regain sight after enough protein has been produced, enough protein from the edited DNA has been produced. So it will depend, it will differ from disease to disease, but some can be very quick. The PCSK9 example, you saw the cholesterol level dropped 7 days after injection, and it just stayed there in the mouse. Yeah, sorry, I think that's a long-winded answer for.
Yeah, for your collateral damage with the Cas13, are structured RNAs less susceptible to the cleavage? So like a tRNA, or are they similar?
That's a really good question. We don't have a very solid answer to that yet. Secondary structure does seem to some extent, affect Cas13 activity. So if it really has a very strongly folded hairpin or something, it may be less accessible. But what we also found is that susceptibility to secondary structure varies from different orthologs of Cas13. Some Cas13s, some members of the Cas13 family from some bacteria are able to target a lot more broadly, while others are much more sensitive. So I think there will be a range, and that's interesting. Yeah. Sorry, you had a question there.
So there's one genome that's very difficult, as far as I know, impossible to edit, that's mitochondrial genome. So do you have any comments or insights on how this can be done?
That's a good question. So gene editing technology has been moving very, very rapidly. So there are two challenges that face mitochondrial gene editing. I think probably the largest challenge is how do you get a gene editing machinery into a mitochondria. Of course, there are a lot of genes that are encoded in the nucleus, and those are amenable to gene editing. But if the mutation is in a gene that's in the mitochondrial genome, then that's harder. And for proteins, you can traffic that into the mitochondria using a localization tag. But for the RNA, guide RNA, we don't really know how to put that in yet. The second challenge is that there are many mitochondria in the cell, and so efficiency becomes a big issue. So how do you edit enough mitochondria to make a difference? So those are the challenges, and that's why it's difficult. Yeah. But the nuclear encoded mitochondrial genes we can certainly edit. Yeah, the one in the black beanie.
Have you thought about, or are you working, or anyone working on? You could use CRISPR to target various activities to specific sites in DNA. Mm-hmm. Have you or anyone working on some way of modifying histones using this technology?
Yeah, a lot of groups have done that. So for example, Charlie Gersbach's group at Duke University has used Cas9 to bring HDAC complexes to specific sites, and that will modify the chromatin. Keith Zhang's groups at Mass General Hospital has taken the TET protein, which alters cytosine methylation on DNA, and that will also change chromatin density or folding. So there are a lot of different ways, and people are exploring that. Yeah.
I'm just sort of amazed that this whole mechanism works. You said you're searching 6 billion letters.
Yeah.
And my question is, how does it do it, and how long does it take?
So gene editing can happen in a cell. You can probably detect gene editing, 6 hours, 8 hours after you introduce the enzymes into the cells. How does it do it exactly? I don't think we have a very clear idea, but it probably is a combination of having many molecules in the cell, so that it's not just one molecule searching through the 6 billion letters. It's a parallel search of many things at once. And then at the same time, there's probably some guidance by the enzyme, because the way Cas9 works is that it first finds a small motif, which is G. Once it finds the motif, then it initiates unwinding of the DNA and then compares the RNA with the DNA. If the RNA and DNA are drastically different, it'll probably fall off and then bounce to somewhere else. That's kind of the general picture, but exactly how, you can probably model that. We haven't done it, but you may be able to do a back of the envelope calculation to see if we're thinking about it right. Yeah. These are great questions.
I have kind of a dual multiplexing question. So I'm wondering kind of at let's say RNA, where you want to edit RNA in a disease process.
Yeah.
And we know so often that, you'll have a whole cascade of genes that will get turned on. So do you think that there's going to be ways to kind of multiplex so you can hit multiple RNAs? And then that kind of on a technological side, on your lateral flow assay, when you're looking for Zika, will you be able to kind of multiplex that as well and look for multiple viruses, let's say?
Yeah, great question. So for RNA editing, certainly our hope is that you'll be able to edit many different sites, even edit many different RNA, but even editing multiple positions on the same RNA. And that can be done by providing the cell with multiple guide RNAs, each one directing Cas13, ADAR to a specific sequence you want to edit. So I think that's definitely doable. And in fact, the way Cas13 works is that naturally CRISPR guides are present in the form of a repeat array. So it's many different guides concatenated together as a single long polyRNA. Cas13 can go and cleave it, chop it into individual guide RNAs. So for editing, you can introduce into the cell a long concatenated RNA, and Cas13 can chop it up and then get directed by each one to edit a specific sequence. On your second question of multiplexing the diagnostic readout, I think that's certainly something that will be feasible. There are different ways to multiplex. On lateral flow, you can imagine having several different bands, each one corresponding to a different thing you're trying to detect. Alternatively, it may also be able to develop a dot array readout where you have a matrix where each spot represents a different thing you're trying to detect. And these are all different ways that you might be able to use the system. Yeah.
So I was finding those the graphs of the amount of papers that have been published recently in these new technologies interesting. And I'm wondering what your thoughts are about this increase in this rapid rate, this exponential rate of papers being published in new technologies, and the fairly stagnant level rate in which the general public is literate about these type of technologies and science in general, and that growing gap. I wonder if you have felt that pressure recently, and how you as a scientist feel like you need to adjust in order to communicate these new technologies efficiently and effectively to the general public.
Great, thank you. This is really a fantastic question. I think it's great that there are a lot of papers being published. I think what that represents is that, one, our ability to conduct studies and our tools are getting better so that scientists can make discoveries faster, can proceed through experiments or multiple experiments using the same amount of time that it takes before. And so our aggregate knowledge that's accumulating is growing exponentially, which is fantastic. It's great for the world, great for developing medicine. At the same time, I absolutely agree with you that we need more education into biology, into bring the biological literacy level up. And that's something that I think all of us have a responsibility to do. Through our conversations with people around us, through our formal and informal obligations to teach and to communicate science, I think those are some of the things that each of us can do our part to make the public understanding of biology better. By giving a community lecture like this, hopefully it will convey some new information about biology to the general community, to the general public, so that our understanding and our appreciation of science can increase, and then people can continue to support the development of science and research. I think those are some of the things that we can all do, but we could all do more too at the same time.
On that note, I would actually love to have a show of hands of who here are non-scientists. Thanks all for coming. And then who would consider yourself a scientist? And what category have I missed? Agnostic. There's a.
Sure, yeah.
The other half of the strand, besides the guide strand, appears to have one or two short hairpin turns and maybe looks like an aptamer. Is that to bind specifically to the Cas enzyme? And would variation of that, like many aptamers, change the way that the enzyme functions? And is that being looked at other than the strand part, which is selective? Or are there things that could be done with the rest?
That's a great question. So the guide RNA has two components. There's the guide, which is what we use to specify what DNA to recognize. And then there's the complicated structure. That complicated structure is just like an aptamer. It's what the protein recognizes to be able to form this RNA protein complex. Now, there are many versions of Cas9, one from each type of bacteria. Each one of them have a different guide RNA, and it recognizes a different structure. So in a way, that's also nice because it allows us to multiplex the system. You can have different Cas9s within the same reaction, and they won't recognize each other's guide RNA, so that you can edit different things, knowing that there's not going to be a lot of crosstalk between these different Cas9 proteins. There are groups that are working on studying what these recognition properties are, how they are mediated. So structural biologists have resolved the crystal structure of different Cas9s bound to different guide RNAs that give us understanding of how the recognition happens. Biochemists have been mutating these RNAs to see how does a single base change in the secondary structure, or how does the change of the entire loop affect the binding affinity of the protein for this RNA. And those are all very useful information for continued engineering of the system.
So then just as a follow-up question, if you were looking at therapeutic opportunities, you'd want to be able to sequence, and that might be, I don't know, on RNA could be 80, 90, 100 pairs or individual letters.
Yeah.
How critical is it as you're making these synthetically, or is it even being made synthetically, that the whole sequence is accurately made?
Very good question. So studies have shown that changes of one or two bases in many positions will probably not affect the binding to that RNA. Maybe it will change the affinity by 5%, 10%, but not substantively enough to be able to make a difference on preventing it from editing the DNA. And sometimes deleting a few bases will still be okay, depending on where you are making the deletion, or inserting sequences will be okay too. So it depends on where mutation is, but certainly there is tolerance for that. And from nature's perspective, that tolerance makes sense too, because if a single mutation is able to completely take out the CRISPR system, then it's not very robust. Mutations happen as cells replicate, and so there is some resilience that's built into the system. Yeah.
I think we'll take one more question. I think we'll go here.
So computers were a lot less accessible to most people back in the day, and then became very accessible to everybody, and now anyone can kind of really just hack and make just about anything out there. Do you kind of, and I'm kind of slowly seeing that more in molecular biology. You can even buy Cas9 CRISPR systems, on do-it-yourself kits and stuff like that online. Just wanted to see what your thoughts are on that, and just kind of that trend.
Very good question. I was wondering when a question like this may come up. It's a very good question. In fact, I was just on a panel a couple of days ago talking about rogue technologies. And we're looking at artificial intelligence, looking at robotics, looking at gene editing. I think these technologies are very powerful. And as they become more easy to use, as they become even more powerful, then how we are going to be using it, I think, requires a lot of thought, requires a lot of sort of thoughtful ways of guiding the use of the technology. It's not a simple, it's not a question that can be answered simply, because there are a lot of complicated issues associated with it. And so the thing that I think we need to do as we move forward is that we need to be careful about what we are doing with the technology, but also be very thoughtful in terms of communicating to people around us how the technology is being used, and also sort of debate and talk about how we should use the technology. I think that the most challenging aspect may come from unintended consequences of a proper desire to use the technology. So, for example, using the system, gene editing system, to engineer a particular species with a gene drive with the hope of eradicating a disease is a very good goal. And if it works, it will have a profound impact, improvement to our health, public health around the world. But there may be, I'm saying there may be because we don't know, there could be or there could not be, there may be unintended consequences too. Because as you engineer a larger system around us, how that system interacts with other complex variables around us to give off other impacts on the environment, those are things that are much harder to predict. So I think that's where we need to be very thoughtful about how we are deploying the technology. It's not the desired use, but really the unintended outcomes that come from a well thought out, well-intentioned use of the technology. Any other questions?
Well, thank you, everyone, and thank you so much
Thank you.
for a really, really stimulating.
Thanks. Thank you.
Feng will be heading up to Keystone. He's one of the lead organizers for our Precision Genome Editing Conference, which will happen Monday through Thursday this week.